back to the synthesis of cholesterol. Already we found that cholesterol is made from acetyl coenzyme A. These acetyl coenzyme A molecules are first used to make mevalonate, then isoprenes, then squalene, and finally cholesterol. Let us see in detail how these reactions are carried out. First of all, two molecules of acetyl coenzyme A react together to produce acetoacetyl coenzyme A. Look here, two molecules of acetyl coenzyme A. The enzyme is thiolase. What is produced is acetoacetyl coenzyme A. Of course, one molecule of coenzyme A is going out. In the next reaction, one more acetyl-CoA is reacting with this acetyl-CoA, producing HMG-CoA. In the first reaction, two molecules of 2-carbon compounds reacted to produce a 4-carbon compound. Now one more 2-carbon compound is added. So now we have a 6-carbon compound. What is HMG-CoA? This carbon is the beta carbon. Beta hydroxy beta methyl glutaryl coenzyme A. So beta hydroxy beta methyl glutaryl coenzyme A is HMG coenzyme A. The next reaction which is being catalyzed by HMG-CoA reductase is a rate limiting reaction in this pathway. Earlier you might have heard that HMG-CoA reductase is competitively inhibited in patients with higher concentration of cholesterol in the bloodstream. Actually, some medicines are there. If you have too much cholesterol in your body, those medicines are applied so that HMG-CoA reductase can be competitively inhibited. What this enzyme is doing? HMG-CoA is reduced using two molecules of NADPH plus H plus. Remember, it is not the NADH which you see in the respiratory pathway or in the citric acid cycle, etc. This is NADPH plus H plus. What happens? Where is the reduction taking place? This carboxyl group is remaining as such. There is no change here. The change is here on this carbon. Earlier it was a carboxyl group. Now it is reduced to an alcoholic group. And of course the coenzyme A is gone out. Now what we have here is a mevalonate. How many carbons are there in mevalonate? You see 1, 2, 3, 4, 5 plus 1, 6 carbons are there in mevalonate. Then this mevalonate is reacting with the ATP. The enzyme is mevalonate kinase. One molecule of phosphate is added here. Again, one more ATP is added to it. Now it is phosphomevalonate kinase. One more phosphate is added here. Now we say 5-pyrophosphomevalonate. This is carbon number 5. So 5-pyrophosphomevalonate. Remember, there is no change in the number of carbon atoms. But in the next reaction, the enzyme is a decarboxylase, which means a decarboxylation reaction is taking place. Decarboxylation is the removal of carbon dioxide. From where? From a carboxyl group. So this carboxyl group is lost in the form of a carbon dioxide. Now how many carbons will be there? Only 5 carbons will be there. What we get is isopentenyl pyrophosphate. Because there is a double bond, it is isopentenyl pyrophosphate. In the next reaction, which is catalyzed by an isomerase, the position of this double bond is shifted from this place to this place. What happens? You see here, we call it dimethyl allyl pyrophosphate. dimethyl allyl pyrophosphate. Remember how many carbons are there? 1, 2, 3, 4, 5. No change in the number of carbon atoms. Then one molecule of dimethyl allyl pyrophosphate reacts with one molecule of isopentenyl pyrophosphate, then one more molecule of isopentenyl pyrophosphate, ultimately producing that farnesyl pyrophosphate, an example for isoprenes. Farnesyl pyrophosphate. How many carbons should be there? 5 plus 5 plus 5. We are supposed to have 15 carbon atoms. You just count and you will see that there are 15 carbon atoms. Farnesyl pyrophosphate. This farnesyl pyrophosphate is reduced using another molecule of NADPH plus H plus. Remember, there was another step of reduction. There also NADPH plus H plus was utilized. Here also NADPH plus H plus is utilized and we get squalene. Can you just find out where is the reduction taking place here? Anyway, you can find it out. What we get is squalene. And how many carbons are there in squalene? How many are there in farnesyl pyrophosphate? That many. And now you can see this squalene is undergoing cyclization. You cannot, from here onwards, you cannot see the structure. You cannot study the structure as such because it is not that much easy. But you can study the names at least. This squalene is acted upon by squalene monoxygenase. Monoxygenase is an enzyme which is adding oxygen. You see here, it is 2,3-oxidosqualene. And this squalene 2,3-epoxide, this compound is undergoing cyclization by what enzyme? Cyclase enzyme. What we get is lanosterol. You need not learn how to draw the structure, but it is better you learn the name of the structure. Just understand that by the cyclization of a long molecule with alternate double and single bonds, we get lanosterol. Thereafter, you see many steps are there. This is not a single step. In several steps, 7-dehydrocholesterol is produced, desmosterol is produced, and these are independently transformed into cholesterol. These are independently transformed into cholesterol. I think it is now clear to you. And this figure indicates how the cholesterol or cholesterol containing compounds are transported in the blood. You see, what you see here is an LDL particle, low density lipoprotein particle. Here, this actually, cholesterol is not that much soluble in water because it is highly hydrophobic in nature. So how can we make it soluble in water? This is unesterified cholesterol here. This is cholesterol ester here. I did not tell you how the cholesterol is being transformed into an ester, but you might have studied in the case of bile salts or bile acids, etc. sometimes. And this is a phospholipid. These phospholipids are having hydrophobic tails and hydrophilic head region. You can see the hydrophobic tail region is towards the inner side, while the hydrophilic head regions are projecting towards the outer side. So what you have here is a molecule which is partially actually soluble in the aqueous medium of the body. LDL particle, low density lipoprotein. It contains cholesterol. And here is the case of atherosclerosis, in which the inner luminal side of the arteries of the heart is reducing in diameter due to the deposition of, you see, LDLs, which contain cholesterol. In this context, often we blame cholesterol, saying that cholesterol will lead to heart attack, etc. But by the time you all might have understood that cholesterol is most essential for our survival. Of course, when its concentration in the body is too much excess than normal, it may lead to unwanted effect. And that is true not only for cholesterol, but for any biological molecule. This is the case of atherosclerosis. It may not be there in your syllabus, but it is good to understand, because it's a field of application, or here a field in which it is important. Here you see this is the innermost layer of the blood vessel called intima. A good lumen is there for the flow of the blood, but as you see here, what happens, this intima is being pushed towards the center, because just outside the intima this plaques are developing because of the accumulation of this LDL molecule. So the lumen, you can see here, the lumen available for the flow of the blood is very much little. It is reduced, leading to heart problems. And major classes of human plasma lipoproteins, you can see here. Chylomicrons are there. They contain free cholesterol and cholesterol esters. VLDL also contain cholesterol esters, but LDL contains the largest amount of cholesterol esters. Okay, so it is always better to have a lower concentration of LDL in the body. And here, what is this? Regulation of the formation of cholesterol. Most importantly, the regulated stage is that of HMG-CoA reductase. While already if sufficient cholesterol is present, this cholesterol by means of feedback inhibition inhibits this HMG-CoA reductase. And here you see cholesterol and other, you know that isopentenyl pyrophosphate is ultimately giving rise to cholesterol, but from the byproducts so many other compounds are produced. You see, biochemical reactions are common, or several of these reactions are common in many of the organisms. For example, in plants, hormones, abscisic acid, gibberellic acids, etc. are produced from this pathway. Carotenoids are produced in this pathway. Vitamins K and E and A are produced in this pathway, and you also know that vitamin D also is produced from this pathway. Rubber in the plants, rubber plants, they are produced. You see, dolichols and quinones and so many types of compounds are produced from this common ancestor. All these are actually indications of a biochemical we say evidence of evolution. All these indicate that there was a common ancestor with a common set of genes, with a common set of biochemical reactions, thereafter fueled by the process of evolution, the diverse types of organisms which we see today are actually formed.